So what are we doing in lecture today? Reproductive. Well, yeah, but are we covering the full chapter today? No. So what are we covering? The male reproductive anatomy. That's right. How many lectures do we have after today? 2 more after today. I know. We're almost there. It's hard to believe it, isn't it? It feels like just yesterday we were starting. I was thinking more about our schedule for lecture because I have it set up for, us to race through all of the male reproductive stuff today. Next week, start out with the female reproductive stuff, and then end with developmental stuff. And then I was looking at the number of slides that I have for reproductive, and it's an awful lot. So I think we're going, instead of racing you through reproductive in 2 days, we're going to take all 3 days for that, which means we won't hit developmental. You should have a class later where you cover developmental stuff anyway, so I don't think this is really going to hurt you guys much. And it really should be a help because that means you only have 2 chapters on your last test instead of 3. So, our goal for today is we're going to cover most of the male stuff, not all of it. So we will be talking about reproductive anatomy, looking at growth structures as well as histology. And when we do that, we're going to be talking a little bit about function. We're going to spend a lot of time on spermatogenesis. And then next time, we'll talk about the hormone stuff. So that'll be next time, and we'll start female stuff next time. And it's going to be a similar deal with the female stuff. We're going to spend a fair amount of time on anatomy, gross anatomy, as well as histology. A lot of time on oogenesis, and then even more time on hormonal stuff because there are 2 different cycles to regulate there with hormones, so it's a little more complicated. Since we have 2 terms on this slide that both have genesis in the name, remind me, what does genesis mean again? Production or formation, exactly. So clearly with spermatogenesis, what's being made? Sperm. And oogenesis? All right. I know we've probably learned it as the egg in the past. In AMP, we don't call it the egg. We have oocytes and eventually an ovum if fertilization occurs. But we'll get to all those points next week. All right, so let's get into this male reproductive stuff. Well, regardless of whether we're talking about male or female, we've got to classify the organs as being either primary or accessory. The primary sex organs are the ones that make the gametes. So generally speaking, we call those the gonads. And the male, that'll be the testes, and the female, that's the ovaries. And gametes is another general term too. So if we were to talk about the specific gametes, it would be sperm or spermatozoans in males, and oocytes and ovum in females. In addition to making gametes, this is also where sex hormones are made. And that's something we hit on when we talked about the endocrine system. So you should already have some idea that testosterone and estrogen are going to be made here. We're going to see exactly where they're made and what they do later. So that's the primary stuff. It's primary because it's most important. Remember, what's the goal of this system? Biologically speaking. To reproduce, to make new members of the species, right? So, anything else that functions is useless without these guys functioning, biologically speaking. The accessory stuff is the external genitalia, any tubing that's going to help the sperm or the oocyte, the egg, to get out of the body, as well as glands with secretions to assist with reproduction. All right, so clearly we already know the testes are going to be the primary sex organ of the male, and we can see one of them on this slide, so you should make a note that they are paired structures. There are 2 of them, and it's this egg-like structure right here. Notice that it's housed outside of the abdominopelvic cavity and basically a sac of integument that we call the scrotum. All right, so that's where the sperm are produced, but the sperm have to be able to leave the body in order for reproduction to occur. So they will first be moved to the epididymis. So they start here, they're moved to the epididymis. And you want to make a note that when they're moved to the epididymis, they're not actually mature yet. So the epididymis is going to store them while they're maturing. Sorry? Exactly. So they're produced in the testes, moved to the epididymis where they're going to mature. And we'll talk about what we mean by that in a little bit. From the epididymis, they're moved into a long tube called the vas deferens or ductus deferens. And notice that that connects to the epididymis, but then it extends all the way up on top of the bladder and then wraps around to the back side of the bladder. And notice on that back side, they're giving it a different label. They're calling that the ampulla of the vas deferens or ductus deferens. You probably want to make a note that ampulla means a widened portion of a tube. Because we're going to see that term again. So when we hit the posterior side of the bladder, we can see that the vas deferens looks like it's fusing with something else, with this other yellow gland right here. So when those two tubes fuse together, that forms our next tube, the ejaculatory duct. And then that connects to a tube that you know. What's the tube that connects to the bladder? The urethra is going to connect the bladder to the external environment, exactly. And you know the regions of that already because we learned those previously. So what's this first region? The prostatic urethra, because it's moving through the prostate gland. When we hit the body wall right in here, that little tiny part, what region is that? We called that the membranous urethra, and that is one term for it. On our figure here, they're also calling it the intermediate urethra, so you could use either term. And then extending past the membranous or intermediate region, we've got what? The spongy urethra. Why is it called spongy? Do you guys remember? Exactly. The tissue looks spongy surrounding it because it's filled with so much vascular tissue. So if you're numbering those off, you probably want to add the numbers that I'm adding here. Because, you know, I always like for you to know how stuff moves through things, right? Yep. It's part of the vas deferens, so I don't really consider it to be a different part. We also have 3 different types of glands that you need to know. 2 out of the 3 are paired. So we've got the seminal vesicle or gland. That's that yellow one that was fusing with the vas deferens to make the ejaculatory duct. We've got the prostate gland, which we've already learned about a little bit. We've mentioned it. And we've got the bulbourethral glands, also known as Cowper's glands. And we'll be talking about their secretions in a little while. Okay, so when we think about the external anatomy for the male, there are 2 different anatomical features. One is the scrotum, which we already said what that was. That's basically a bag of integument that holds what? The testes, exactly. But how do we classify the testes again? Are they accessory or primary? They're primary, so they're the most important organs in the system. Doesn't it seem kind of weird that we hold them outside of the abdominal pelvic cavity then? They're kind of in a vulnerable place. So why? For temperature control, because sperm cannot survive or cannot be produced at body temperature. So by keeping them out of the abdominal pelvic cavity where we have all these organs moving around and creating body temperature, they're held in a place where heat can be lost to the external environment that cools them off by a few degrees Celsius. But, thinking about that external environment, the temperature there can change a lot, right? So there's got to be some way to regulate exactly how much the testes are being cooled off. And that's done with two different types of muscles. So we've got the dartus muscle and the cremaster muscle. Dartus causes the skin of the scrotum to wrinkle, whereas cremaster is going to regulate how close the testes are held to the abdominal pelvic cavity or up by the body. So when would we want these guys to contract? When it's cooler or hotter outside? When it's cooler outside, exactly. What will wrinkling the skin do? Well, that's what the cremaster does, right? The cremaster helps with deciding how close they are to the body. So what will wrinkling the skin do?
Keep them closer together. Less surface volume. Thicker.
Makes it thicker. In other words, it provides a thicker layer between the testes and the external environment, so it's kind of like them putting on a winter coat. So clearly, both of these muscles would have to contract when it's cooler. Cremaster, bringing them closer to the body, is going to help them to be exposed to more of that warm body temperature so that they don't get too cool. If we look inside the scrotum at the testes, we can see, well, there's some other stuff that we're going to have to know. Because when we look inside of them, we can see that the testicle here is filled with tubes. Those are the seminiferous tubules. And notice they're highly organized, just like we've seen with other tubes and other organs. So it looks like they're kind of chopping up the testes into small chunks or lobules. And look at how many there are. Up to 300 lobules per testicle. So it's not enough to know all of the major structures that I numbered off if I ask you how the sperm get out of the body. You got to be more specific because there are other smaller tubes in here. So they are produced in the seminiferous tubules, specifically in the testicles. And then they're moved through the straight tubules to the rete testes, which connects to an efferent ductule. What did efferent always mean? Exits, exactly. So those are the tubes that are allowing the sperm to leave the testes and enter the epididymis. And once in the epididymis, they're in the ducts of it. We can even take a closer look at the setup here. So we have a microscopic image where we've cross-sectioned some of the seminiferous tubules. We can see one full tubule here. And then on either side of it, we see portions of others, some of its neighbors. But then there's other tissue in there, right? There's tissue filling in between the tubules where the sperm is being produced. So what you're seeing in the wall of the seminiferous tubule, these are actually the cells that are going to become sperm, for the most part. And then between the seminiferous tubules, we have these interstitial cells also. Notice that they're called Leydig cells sometimes, so either way. And we care about those cells because those are the cells that make androgens, sex hormones. Yeah.
Could you repeat that last place? Which ones are the ones that make the
So the interstitial cells or Leydig cells, they're the ones that make the androgen. So here that would be the testosterone. Oh, and also, guys, when you read this chapter, they go into a lot of detail about, for example, that there are different types of estrogens. We're not going to go into those specific details, okay? But you will need to know things like the function of estrogen and the functions of testosterone. We talked about blood supply in a lot of organs, so it shouldn't be surprising that you're seeing it here. These are actually vessels that you studied in lab, except when we learned them in lab, we called them gonadal arteries and veins. So now that we're specifically looking at the male, they're testicular arteries and veins. And those guys are bundled up with the vas deferens and other tubes like lymphatic tubing and nerves into something that we call the spermatic cord. And so if we look back at that picture where they're showing us the epididymis and one of the testes, we can see right at the top here, as the vas deferens moves away from the epididymis, it gets bundled up with other things. This is one point where people were having trouble in some of my earlier classes, so you probably want to make a note that the spermatic cord is not just this outer tissue here. It's that tissue plus all of the stuff that it's bundling up together, including the vas deferens. In lab, you're going to have to find a lot of these structures today. So when you're looking for the vas deferens, do not look for it in the spermatic cord. It's going to be covered up with all of the blood vessels there. Instead, follow the spermatic cord up toward the bladder, and you're going to have an easier time finding a place to label the vas deferens. All right, the other external genitalia is obviously the penis. Define it as the male copulatory organ. And we've mentioned previously that it's filled with vascular tissues. Here, I want you to know that there are three areas, three compartments of vascular tissue. So we have one area that specifically surrounds the spongy urethra, and that's why we call it the corpus spongiosum. It basically has spongy urethra in the name there, right? Because it starts with spongy for spongiosum. And then toward the anterior portion, we have the corpus cavernosum. However, there are two areas of that, so there are two of those. And when we talk about them together, we call them the corpora cavernosa. So singular, it's corpus cavernosum.
Why is there blood plural?
Because there are two of them.
Oh.
So there's only one corpus spongiosum, but there are two areas that we call corpus cavernosum. And that'll make a little more sense when we look at some of the plastic models in lab, so that we can see a cross-section where we can see all three of those compartments. All right, so in order to assist copulation, the penis must form an erection, and in order for this to happen, those vascular spaces, corpus spongiosum and the corpora cavernosa, must engorge with blood. So we know about the external features. We're going to move into the tubes, the ducts next, and we're going to start with the epididymis, since that's the next place the sperm are headed after they're made in the testes. So, in the epididymis, I want you to know that the epithelium there has microvilli. That's important. We've seen microvilli before. What do those always do for organs? Microvilli, every time. They increase surface area. Why? What? To help with absorption, exactly. And notice it's similar here, right? They're helping to absorb some of the fluid that the sperm are bathed in. However, they don't just absorb stuff, they're also going to be adding nutrients to that fluid. Why would we need to add nutrients to the fluid here? To keep the sperm alive, exactly. Sperm are living cells. All living cells need nutrients in order to make ATP. And sperm can be stored in the body for months. So it's really important to be giving them nutrition here. All right, we said that the sperm were immature when they enter the epididymis, so that means that they cannot move on their own. But by the time that they leave the epididymis, they are capable of moving on their own, so they're fully mature. Mm-hmm. Yep, it stores them until they're mature. Even though they are capable of moving on their own, in order to do a mass movement and get them to move a longer distance in the tubing here, because there's a lot of tubing, right? From the epididymis, where are they headed next? The vas deferens, which was a really long tube, right? It went all the way up and over and around the bladder. And then there's also ejaculatory duct and the urethra, which is another long tube that they're going to have to move through. So to get them through that tubing in a decent amount of time, the tubing has to contract. So all of the tubing here has smooth muscle in it, in its wall. And what do we call those contractions when the smooth muscle contracts to move stuff toward the outside of the body? Peristalsis. Very good, just like it was in the GI tract. All right, I already told you that sperm can be stored in the body for several months, but if they are not removed from the body in that amount of time, then they'll be phagocytosed because they should be dying off at that point. All right, I already told you that the rest of the tubing has muscle in order to move the sperm out of the body. But I did put this other point in here about vasectomy because I think it's kind of interesting. Notice it starts with vas, just like vas deferens. Ectomy means cutting. So with a vasectomy, the vas deferens gets cut, and the two ends on either side of the cut get sealed off. This is a form of birth control, which is highly effective. However, men are advised to not have unprotected sex for 1.5 to 2 months because that's how long it takes to get all of the sperm out of the tubing past the cut. All right. From the vas deferens and ejaculatory duct, we know that they'll be entering the urethra, and you already know that the urethra sometimes holds urine and other times will hold the sperm with the semen. You also already know the 3 regions of it. So which region do they hit first? Prostatic, followed by membranous or intermediate, followed by spongy. That's exactly right. So that's it for the tubes. Moving on to the glands. We'll start with the seminal vesicles, just because they're the first secretions that the sperm will come in contact with. It's really important to note that the secretions here are alkaline. So what does that tell us about the pH? They have a basic pH, so a pH greater than 7. Why is that going to be important? It protects the sperm. What does it protect the sperm from? Potentially from any urine that might be left over in the urethra. Also from something else.
The acidity in the vagina.
The acidity in the female reproductive tract, exactly. Exactly. So remember, the whole goal as far as this system is concerned is to make a baby. So the sperm have to be protected from the acidity in the female reproductive tract. Besides being basic, it is important to note that there are some chemicals included here that will help to activate the sperm so that they'll swim or enhance that swimming. You also should know that most of the volume of the semen comes from the seminal vesicles, so 70% or so. And when it comes to glands, we do need to note where all of them will be secreting to. Remember, these guys have tubing that connects to the tubing of the vas deferens, right? And when that happens, what do we call the tube that's formed? Right, because we have a seminal vesicle here, and its tubing is connecting with the tubing of the vas deferens. So this point here where they come together, what do we call that? The ejaculatory duct, exactly. So make a note that the seminal vesicles secrete to the ejaculatory duct. The prostate gland has secretions that are a bit acidic, but that's okay because they're not contributing the most volume to the semen, so the semen will still be basic. The reason why they're a bit acidic is because they have some nutrients that are acidic, like citrate. And they also have factors that are going to assist with the sperm swimming, helping to activate the flagella so that it will move. Even though there are two different types of tubes in the prostate gland, right? Because we've got the ejaculatory duct over here, and we've got the urethra coming through it as well. The prostate gland will only secrete to the prostatic urethra. And don't let that point trip you up when you're trying to label the different types of tubes in the prostate gland. So look at your connections. You know the urethra has to connect to the bladder. Yeah, Hyatt?
So when the sperm was immature, did that mean it didn't have the flagella yet? Or it did, but it wasn't adequate?
It has the flagella, but they're not fully functional yet. And you'll actually be seeing that because next week we're going to look at some histology. And so you're going to get to look into the seminiferous tubules. And when you do, you're going to actually see the flagella of the sperm in there. All right, our last glands, the bulbourethral glands, are responsible for producing pre-ejaculate, which does a couple of things. It acts as a lubricant, so it's a more viscous solution. It also is basic, and the whole reason for it is because it's going to help to flush most of the leftover urine out of the urethra so that the sperm are not exposed to the acidity of the urine. Okay, so we know everything, right, that is helping to make the semen. We know about the sperm, we know about the secretions of all three of our glands, but I want us to take a closer look at some of the components here. So there are a lot of nutrients, not just fructose, there is also some glucose. There are also a number of things that assist with sperm movement. You can make a note that relaxin is one of those things. An interesting component is a class of chemicals called prostaglandins. And they actually do a couple of interesting things. So on the slide, it's telling us that they're decreasing viscosity of mucus, right? So the cervix is actually the first part of the uterus that the sperm have to get through as they leave the vagina. And the cervix has an opening that's filled with the mucus that's really thick. That can vary depending on where the woman's at in her menstrual cycle, but it tends to be a thicker mucus. So prostaglandins actually help to liquefy or make that mucus more watery to make it easier for the sperm to get into the uterus. Not only do the prostaglandins do that, but they also cause reverse peristalsis to happen. Why would it be called reverse?
Maybe it makes the uterus go.
Well, the sperm is only to the, like, go back in.
Exactly, because it's contraction of smooth muscle still. That's why it's peristalsis. However, it's not moving stuff out of the body. It's helping to move stuff further up into the body. In other words, the prostaglandins here are causing the uterus to contract in such a way that it's creating a current to help pull the sperm higher up into the uterus, and that's important because if fertilization is going to occur, it has to occur in the fallopian tubes that are attached higher up in the uterus. Other stuff. We already know semen is basic or alkaline, and we have talked about why. Why would it need factors that are immunosuppressants?
To reduce the immune response to the magnetic cell uterus.
Or even more importantly, for the sperm, right? Because the sperm are foreign cells that have been given to the body. So as far as her immune system is concerned, that's stuff to attack. Those are foreign cells. So we have chemicals here to suppress that immune response to keep the sperm alive. However, that could cause some problems, right? Because if her immune system is dropped here, what could happen? If the immune system function has gone down, what might happen?
She could get an infection.
Infections, exactly. Bacteria might be delivered along with the sperm. So we also need to have antibiotics or antimicrobial agents as part of the semen to protect the reproductive environment from infection. One last thing, which is also kind of interesting, there are clotting factors that will cause the semen to coagulate and stick to the side, the inside of the uterus. And that's important because that's going to make sure that the sperm stay in the body. Fibrinolysin is another chemical that will be activated later, which will liquefy this clot in order to release the sperm so that they can swim up the fallopian tubes. All right, so that's all of the big stuff. We need to talk about spermatogenesis for a bit, though. So we already know that sperm production. We already know where it happens in the seminiferous tubules and the testes. But we got to review a little genetics. This should be review stuff from general biology. All of the cells in the body besides the gametes are 2N. They're diploid. What that means is that they have 2 copies of every type of chromosome. So one copy came from mom, another copy came from dad. So we could say that, like, you got one copy of, say, chromosome 1 from mom, chromosome 1 from dad. That's considered a homologous pair of chromosomes because they carry the same types of genes. So, like, if chromosome 1 carries the gene for hair color, you have one gene that came from mom and one gene that came from a chromosome from dad. So total, in human body cells, we have 46 chromosomes, 23 of which came from your mom, 23 that came from your dad. When gametes are produced, we have to cut that number in half. So the gametes will only be haploid. They'll have one copy. We're going to be comparing mitosis and meiosis, but I know I'm going to run out out of space on that slide. So let's make a little note here about some things that we're going to be talking about. When we talk about chromosomes during cellular replication, we often think of them in this X kind of shape. After the DNA has been condensed, and we have to know the names of those parts to understand what we're going to be talking about. So each line that I drew here represents one sister chromatid. The dot in the middle is something called the centromere. So they're proteins that are essentially sticking our two sister chromatids together. And what the sister chromatids are is one copy of original DNA, and then the other one is DNA that was replicated from that original one. So both of those lines hold the exact same genetic material. So that overall X-like structure, this whole thing, is the chromosome. When we talk about meiosis, I'm going to be using this term tetrads. Tetra means 4. So one of the differences between meiosis and mitosis is that in meiosis, we don't just have the individual chromosomes hanging out by themselves. Instead, we stick them together with their homologous pair. That forms the tetrad. So, for example, we could take that original one that I drew, say that that's all genetic material from Dad, and then we can stick it together with the genetic material from Mom. And each one of those X's still represents the same thing that I drew in the bottom corner over here. It's just now we've stuck all of the traits for, say, hair color together in the same place. Does that make sense? Yeah? Okay. All right, then let's talk about meiosis versus mitosis. What do we always start out with? A cell that's haploid or diploid? Don't guess. So what did haploid mean again?
One sister cell?
Sorry? What did the term haploid mean?
One sister cell?
Half the amount of genetic material. It wouldn't make sense to start out with a cell that was haploid, right? So regardless of if we're talking about meiosis or mitosis, we always start out with the cell that's diploid, 2N. With mitosis, what are we making?
An exact copy of the same cell.
Exactly. So with mitosis, we're forming two cells that are essentially clones of the one that we started out with. So we have two cloned daughters here. So they hold the exact same genetic material as each other and as the cell that we started out with. With meiosis, it's a different story though, right? So with meiosis, first off, how many rounds of replication do we go through? 2 different rounds, that's right. And we specify those with Roman numerals. So we have the first round going on here, and that's going to create 2 new cells. Depending on your book, sometimes they'll call those cells 2N, sometimes they'll call those cells 1N. So I'm not going to ask you if those cells are haploid or diploid, but you do need to know that we have produced 2 unique daughters here. Unique because they hold different genes. They both have the same amount of genetic material as each other, but they hold different genetic material. And they're going to go through the second round of cell division, ultimately leaving us with 4 cells at the end of the process. And each one of those cells is going to be haploid. Not only are each of those cells haploid, but they're all unique. And remember, they're all unique because each one has different genetic material. So, when would we see mitosis happening?
In body cells,
But why? Why would we see cells going through mitosis?
For repairing.
For repair, for sure. Why else? To replace cells, because we've talked about how most of the cells in your body don't last a lifetime, right? The neurons do in the nervous system, but everybody else pretty much has to be replaced at some point, like the red blood cells that we talked about. So yeah, to replace, and also during growth and development. And mitosis can happen almost anywhere, right? Okay. Meiosis clearly is only going to happen when? For reproductive purposes, to make what? To make the gametes, the sperm and the egg. And if you ever have trouble remembering mitosis versus meiosis, you can always remember that mitosis can happen anywhere, like in your little toe. Meiosis starts with me because it helps to make a little me. That's when a student taught me. All right. We have to look at the phases, so let's do a little refresher. The whole point of doing this is so that you understand spermatogenesis better in just a minute. So let's focus on mitosis first. What are the phases of mitosis? We got prophase. Good. What else? Metaphase. Anaphase. And telophase. Good. So you can always remember that as PMAT, right? We need to talk about a few highlights that happen in each of these steps. Now, before prophase ever happens, what have we got to do to the DNA? We've got to replicate it. So DNA replication happens before prophase. And then during prophase, we're going to do something else with that DNA. What are we going to do to it? Not in prophase. Remember, DNA is one of our macromolecules, right? Like a huge string. So what do we need to do to it? We've got to condense it. We've got to condense it. So let's make a note that the DNA condenses here in prophase. And so it's basically the exact same effect as like working with fishing line. If you've ever seen fishing line at the store, it's wound up around a little spool, or like yarn, it's always wound up, right? Or thread. So that way it's easier to move around. It's a similar deal here with the DNA. What is actually going to be moving the DNA around? Because they don't have legs, they can't move themselves, so what's going to pull them around? The spindle. The mitotic spindle has to form during this phase then. So let's make a note that the spindle forms. But when that mitotic spindle forms, where does it form? Does it form in the nucleus or the cytoplasm? In the cytoplasm of the cell. And that's kind of a problem then, right? Because we have this spindle outside of the nucleus, but the DNA is inside the nucleus, and the two have to be able to interact. So what else needs to happen? The nuclear envelope disappears or degrades. Degrades, good. And I'm going to have to use shorthand so I don't use up all of the space here. Metaphase, there's one key thing that happens during metaphase. What happens? The chromosomes align along the middle. Good. And then during anaphase, what's going to happen? Who separates? The sister chromatids separate, so we degrade the centromere. Right, we break up the centromere so that we can separate the sister chromatids. And when they're separated, we actually pull them to opposite ends of the cell. All right, telophase. What happens during telophase? Well, this is the last phase, right? So what's got to happen? Yes, we've got to break up the cytoplasm and all of the organelles to form two new cells. Two new cells. So, does anybody remember the name of that process? It started with a C. Cytokinesis. Very good. So, cytokinesis, or splitting of the cytoplasm, happens here during telophase. And then we kind of have to put the cells back together so that they look like the cell that we started out with, right? Because they're clones of it. So I always think of telophase as also being just the opposite of prophase. Because we're going to have to decondense the DNA so that it can be read. We have to reform the nuclear envelope. And will we need that mitotic spindle anymore? No, it's just taking up space. So we degrade it. Any questions about mitosis? All right, then we've got to talk about meiosis. The nice thing about meiosis is that we have phases with similar names, right? We still have prophase, metaphase, anaphase, and telophase, but we distinguish those with Roman numerals to tell us if we're in meiosis 1 or 2. I'm not going to go through every single little thing that happens in each one of these phases, but we do need to compare the phases of meiosis to the phases of mitosis. And the ones that are most different happen during meiosis 1. So before meiosis happens, are we going to see DNA replication? Yeah, we do. So before prophase 1, we still have DNA replication. And then during prophase 1, we still have the DNA condensing, the spindle forming, nuclear envelope degrading, all of that same stuff as mitosis. But there are two different things that happen. One of those things is forming those tetrads that I drew for you 2 slides back, right? So we have a term for that. We call that synapsis. Can anybody tell me what the purpose is for doing this? For sticking the homologous chromosomes together?
Is it so they can switch?
Exactly, so that they can switch out some genes. That's the only way we're going to get 4 unique cells at the end of this. Because right now, we have our first 2 blue sister chromatids with the exact same DNA. And then the 2 red ones have the exact same DNA. So if we just left it as is, at the end of this, we'd have 4 cells still, but 2 of them would have the same DNA, and then the other 2 would probably have the same DNA. So they switch out arms. So we call this crossing over. And the reason why this is okay is because they should be holding similar genes at similar points. And that helps us to create 4 unique sister chromatids now that we can split up. Right? Because we have the first one that's still DNA all from dad. The second one that's mostly DNA from dad, some from mom. The third one, which is mostly DNA from mom, some from dad. And then the fourth one that's all DNA from mom. Any questions? No? This should be review. All right. So those are the important differences with prophase 1. There's an important difference with metaphase 1. How's that going to be different? What happens during metaphase 1? Well, you still have the genetic material aligning along the middle. But is it the individual chromosomes that line up along the middle? It's the tetrads instead, right? Because we stuck our homologous pairs together. So tetrads now align along the middle. And then during anaphase 1, what's going to happen? The tetrads split apart. And so then instead of sister chromatids being pulled to either end of the cell, we have whole chromosomes moving to opposite ends of the cell. Telophase 1 is, that should be a 1. Telophase 1 is a lot like telophase and mitosis. However, some of those steps might not fully complete, like all of the things that were opposite of prophase. Depends on how soon we start meiosis 2. One key difference that happens here at telophase 1 is what you end up with. Because what do we make? We make 2 cells, but how is it different?
They're unique.
Yep, they are unique. Prophase 2 is a lot like mitosis, but one really important difference is that before this prophase happens, there will be no DNA replication. Metaphase 2 is similar to mitosis. You have the chromosomes aligning along the middle. Anaphase 2 is still the same as well because the centromeres are going to split so that we can pull the sister chromatids apart. But by the end of telophase 2, what have we created? 4 unique cells that are all what? Haploid. That's important. So questions about any of that?
For the telophase 1, they're making 2 diploid or haploid.
Well, that was the point where I told you that some books will call that diploid and some will call it haploid. And if you really want to know the answer as to why, we can talk about that during our break between lecture and lab, because that's a little higher level concept, okay? So I won't ask you if the cells produced here after T1. I won't ask you if they are 2N or 1N. But you do need to know that they're 2 unique daughter cells. All right, well, all of these purple slides are on my notes on meiosis versus mitosis. So you have those for your own reference. But we still need to spend some time talking about spermatogenesis. And notice, spermatogenesis doesn't start out with meiosis. It starts with mitosis. So we're going to see stem cells, the spermatogonia here, going through mitosis to clone themselves. And part of that is going to be forming spermatocytes. The spermatocytes are going to go on to go through meiosis. But notice, at the end of meiosis, they still haven't formed true, full, mature sperm yet. They've formed these spermatids. So the spermatids are going to have to go through a third process, spermatogenesis, where they mature and become full sperm. So that's not cellular division. That's more like a modification to the cells that are made. And spermatogenesis sounds a lot like spermatogenesis. So I will always remember that spermatogenesis involves the spermatocytes, whereas spermiogenesis is where we're actually making the mature sperm. And luckily, they give us a picture to help us kind of visualize what happens here. So, what are we looking at? We are looking at part of a seminiferous tubule. So we have the outside of it here. We have the inside with the lumen down here at the bottom. And they're showing us cells in the wall. While we're here talking about the process of spermatogenesis, kind of think about this as them showing us the cells that we start out with plus the cells that they are making. So we start out with the spermatogonium, that's the stem cell, and we said the stem cell is going to go through what process? Mitosis, exactly. So out here where we find the spermatogonia, notice we're calling that the basal compartment. That's the only place where mitosis is going to happen. So the spermatogonium is going to go through mitosis, and when it does, it forms 2 different cells. We call one a type A and the other a type B. And notice what's happening with the type A. The type A cell is staying right up by the outer edge of the seminiferous tubule, so that it can become a new spermatogonium. So this type A cell is replacing the one that we divided. The type B cell is going to move down into the next compartment, the adluminal compartment. And when that happens, we change its name. We're going to call that a primary spermatocyte.
Professor,
Mm-hmm.
so the type A cell is the old one, and the type B is the new one?
Well, but the old one already split, and that formed two new cells.
Oh, okay.
Yeah. So those are the two cells that are the clones of the old one. So the type A is going to go back and replace. It's going to stay at that outer edge and become a new stem cell to replace the one that just split. Otherwise, we'd run out of stem cells, and then they wouldn't be able to make more sperm. So about that type A and type B cell, would they be 2N or 1N?
2N.
They both are 2N. That's exactly right. Also, we said that they're clones, right? They're not unique. They are the same as each other, and they're the same as the stem cell that we started out with. Okay, but our type B cell is moving into the adluminal compartment. There it becomes a primary spermatocyte, and the primary spermatocyte goes through the first round of meiosis. When that happens, it forms secondary spermatocytes. Which, are they the same or are they different? They're different. They're unique compared to each other. They're also unique compared to the cell that we started out with here, the primary spermatocyte. And the secondary spermatocytes go through the second round of meiosis to form spermatids, and we know those spermatids are definitely haploid. They also are all unique. But if we look at them, man, they don't look anything like sperm yet. So once meiosis is done, there's still some work to do to turn these guys into true, full-fledged spermatozoans. I gave you this slide to help you remember where things are 2N or 1N, but remember, because scientists don't totally agree on these guys, I'm not going to ask you if the secondary spermatocytes are 2N or 1N. You do have to know that two of them are produced from meiosis 1, and you do have to know that they are unique. All right, so what goes on with this spermatogenesis? Well, clearly we have to change the structure of these cells quite a bit to turn them into sperm. And that's going to take us about a month. So it takes about 24 days or so for a spermatid to turn into a true full-fledged sperm cell. You do not have to memorize every single little step here, but there are some important things that I want you to note. One of those things is that a flagellum is going to grow. Another is that the cytoplasm, a lot of the cytoplasm anyway, is going to be lost. So a lot of the cytoplasm sloughs off into the surrounding fluids, and that's to help streamline these cells. By the time we're done, we've also rearranged the organelles, and we've created 3 different regions: a head, midpiece, and tail. So those are regions. The structure within the tail is the flagellum, right? What would we find in the midpiece? What organelles are they showing us there? The mitochondria. Exactly. Why does it make total sense to stick the mitochondria right next to the flagellum? To make ATP, to give it the energy to move. And what do we find in the head? The nucleus, the genetic material, the DNA's in there. And that's not the only thing. Notice it looks like it's got a little cap, the acrosome. We care about the acrosome because it's filled with enzymes that are going to help the sperm penetrate the egg so that it can deliver its DNA to the egg, or oocyte. And I gave you the wording for all of that stuff on this last slide. So that's where we're going to leave it for today. We're going to pick up here with other cells that we find in the seminiferous tubule when we come back next week. And then we'll talk about male hormones and start to talk about female reproductive stuff. If you have questions, let me know during the break. Otherwise, you guys are free to move around and wake yourselves back up.